I would like to proceed to the last presentation in the session. This is given by Christy Hawkins from Caltech, and she's going to speak about metabolic engineering in Saccharomyces.
My name is Christy Hawkins. I'm a graduate student in Christina Smolke's lab, and the work I'm presenting today is a little bit different from that you saw from her talk yesterday. In the spirit of synthetic biology, I've renamed my talk from something a little more long-winded to yeast alkaloid factories, since what I'm presenting today is really just another example of microbial cell factories that we've been seeing all weekend. So the specific pathway that we were targeting is a class of benzyl isoquinoline alkaloids. These are secreted from the lactocophers of opium poppy and other BIA-producing plants. The most well-known of these are morphine and codeine, which are widely used analgesics. But there are actually over 2,500 different elucidated structures of various BIAs. And because of their natural role in plant defense, many of these possess therapeutic properties that we can take advantage of. In addition to the analgesics, there are also many antimicrobial agents and even very promising anti-cancer and anti-HIV therapies. Because this pathway is so important in plants, the enzymes are very well characterized, and a lot of these activities have been cloned from the plant host, largely with the goal of either doing metabolic engineering strategies in plants or transferring those to a recombinant host, although very little work has been done so far to this end. So we've heard a lot of the reasons why we like to use yeast as a production host. It has a very extensive history as an industrial workhorse. It's a grass organism. It allows green synthesis or white biochemistry, avoiding the harsh reaction conditions and toxic waste streams often associated with organic synthesis. And of course, it's easy to do genetic manipulation. There's plenty of plasmid-based vectors that you can use, as well as the ability to do targeted chromosomal integration, which is a major advantage. So why specifically do we want to turn our yeast into an alkaloid factory in this case? I mentioned that some people would like to do metabolic engineering in plants to overproduce these compounds. However, since the secondary metabolism is very complex in plants, the metabolic engineering strategies, such as RNAi, often elicit very unpredictable results. So it's very difficult to do any type of forward engineering. Also, purification from plants is very rigorous and can also be very costly. A major reason why we chose a eukaryotic host for this work is that it allows functional expression of membrane-associated proteins, specifically cytochrome P450s, which are scattered throughout this pathway. And although it's not perfect in yeast, you have a much better chance than getting it to work in bacteria. Also, transferring this pathway to microbial host allows the production of rare intermediates that normally don't accumulate to any level in the plant, and also the ability to recombine enzymatic activities to produce non-natural alkaloids, which they are very interested in doing, specifically with the Bain and making a lot of derivatives of that. You can do it all in one synthetic host if you're expressing the entire pathway. Okay, so this is just a very simplified diagram of the BIA pathway and the various metabolites that you can produce. So all BIA molecules are derived from two molecules of tyrosine. One goes to dopamine and one to 4-hydroxyphenylacetaldehyde. The condensation of these two molecules produces norcochlorine, which is the first committed step in BIA biosynthesis. Norcochlorine then undergoes a series of methylation reactions and a 3' hydroxylation to produce S-reticulene in plants. The S-reticulene isomer can go to sanguinarine and berberine, and these typically have antimicrobial properties. However, the S isoform can also be converted to the R form to move down the morphinium branch. So one of the first things that we were able to demonstrate is reticulene production in yeast using the commercially available substrate norlon-noseline. So since the only difference between norlon-noseline and norcochlorine is the presence of a 3' hydroxyl group. So since this is normally added by CYP-ADP1 in the native pathway, we're able to actually get the correct product, reticulene, using only 3 methyltransferase activities: a 6-O-methyltransferase, an N-methyltransferase, and a 4'O-methyltransferase. We're able to characterize our products using LCMS and MES, which, expressing each enzyme individually and in combination, we're able to determine the methylation occurs in the correct positions by the characteristic fragmentation pattern. So we were fortunate enough to have enzyme variants for each of these activities available from both T. flavum and P. somniferum. So we wanted to combinatorially test these using a plasmid-based expression system to see which combinations were best for our strains. So we found 3 that we chose to make stable integrants of. And we found that in general, the T-flavum CNMT gave us lower productivity overall, which according to individual enzyme studies, it actually prefers smaller isoquinoline substrates. So this is in line with what we would think. So once we selected our stable strains and made chromosomal integrations, we wanted to see if we could further tune our enzyme expression levels so that we wouldn't be overexpressing enzymes that we didn't need to express. So we did this using an engineered Gal promoter system. So what we did was we titrated each enzyme individually by expressing it from the P-Gal promoter while holding the other two enzymes constant from the constitutive TEF1 promoter. We did this in a Gal2 knockout background, which we previously showed gave a more linear and homogeneous induction response with galactose concentration, so you can more finely tune enzyme expression. With this system, we were able to generate curves that establish metabolite production as a function of galactose concentration. You see here we're looking at the extracted ion chromatograms of reticulene, and we get an increase in reticulene production with increasing Gal. And then we're able to generate curves relating that to each of our enzymes. But we didn't want to use an inducible promoter in our system because obviously that's not ideal for an industrial process. So we wanted to relate that back to a constitutive promoter. So we used the TEF library generated by the Stephanopoulos group at MIT. And we found that in most of our strains, we can make the promoter substitution to TEF7, which only has about 16% of the expression level of the native TEF1 promoter. And we see here in 2 of our strains, we actually get slightly better production after 48 hours compared to the original constructs containing the TEF1 promoter. However, in one of our strains, we noticed that we couldn't titrate the T-flavum 6-OMT enzyme and still generate a curve in this range. And we found that when we made the promoter substitution, as predicted, we took a huge hit in production. So that tells us a little bit about this particular enzyme variant, that it requires higher expression levels to compete. Okay, so once we had established reticulene production, again, it's not fully optimized. We still found that transport was limiting, and also with norlaenostrine not being the native substrate, we kind of take a hit in our enzyme activity. But we felt that we can move on to other downstream branches. So one of the ones that we wanted to target are the sanguinerine and berberine branches, which again are antimicrobial agents. Okay, so from S-reticulene, the first reaction is berberine bridge enzyme to produce scolurine. This is a unique bridge-forming reaction that cannot be duplicated by synthetic chemical means. From there, in the sanguinerine branch, the initial enzyme activities have not yet been cloned, but we were able to make significant progress down the berberine branch. Using a scolurine 9-O-methyltransferase and a P450 to form this methylene dioxy bridge, we're able to accumulate tetrahydroberberine, which is only one step away from berberine, the final product in this branch. However, this final enzyme activity remains to be cloned. So again, we used LC-MS analysis to determine the correct production of each of our metabolites, and we found that all of these reactions occurred correctly in our yeast host. So for the first step, we see very good conversion to scolurine and to tetrahydrocolumbamin, almost full conversion. But we take a huge loss at this final step, which is the P450 activity. And the best we're able to do so far has been about 10% conversion. And our best strain has an integrated copy of the Arabidopsis ATR, which is the NADPH P450 reductase. We also found that this enzyme was not active with human or yeast CPR variants, but it is active with the more recently cloned P. somniferum CPR. So there's something specific about the plant CPRs that is required for this particular enzyme activity. Okay. So another branch that we wanted to target was the morphinium alkaloids. Obviously, this is going to be very valuable as a therapeutic. So in plants, only the S isoform is initially produced of reticulene, and then it has to undergo 2 additional reactions to be converted to R-reticulene. And these enzymes have been identified, but they have not yet been cloned from the plant host. But in our system, since we're feeding a racemic mixture of RS norlaenostrine, we also are able to produce the R form of reticulene, enabling us to move down this morphinium branch. So the first activity we tried was a salutarity synthase activity. But since this enzyme had not been cloned at the time of our work, we used a novel human CYP2D6 activity. So this was believed to be implicated in this reaction because human white blood cells make a small amount of morphine, but it had not yet been shown experimentally. We were able to demonstrate this reaction in our yeast host. And again, we show the correct fragmentation pattern of salutaridine, but activity is somewhat limiting here. So we wanted to do a little bit of optimization on this P450 and its reductase partner. So we found that using the native sequence, we only get about 7% conversion of reticulene. Codon optimization does not give us much benefit at all. Integrating the human CPR reductase gives us a little bit of a bump, but ultimately we had to express this from a strong promoter and a high copy plasmid to really get much activity out of it. And other variants using a soluble yeast CPR were somewhere in between. So this tells us that the activity is really what's limiting in this case. And this is not terribly surprising since human P450s are notoriously promiscuous, meaning that they have a broad substrate specificity but low activity on all substrates, and that's what we're observing here. So from there, we wanted to proceed down this pathway and make additional morphinium alkaloids. So while we were working with the human P450, the sequence for salutaridine synthase was published, although its characterization has not been published. So this is a STEP719B. So we were hopeful that this would actually outperform our human variant. But so far, we have not been able to really optimize this activity. We tested a number of reductase partners, and we're not seeing accumulation of salutaridine, although we see enough to push us to more downstream products. We feel that this is still limiting. And surprisingly, it's not functional with the P. somniferum CPR that has been cloned, since this enzyme is also from P. somniferum. But since plants have multiple CPRs, it's possible that this is still not the correct reductase partner, even though it comes from the same organism. But from salutaridine, using both variants, we were able to produce, we were able to also clone SAL-R and SAL-AT, which is an acetyl-CoA-dependent reaction to produce salutaridinyl 7-O-acetate. This is directly converted to thebaine, which is reported to be spontaneous at pH 8 to 9. However, in plants, they've also detected the presence of a thebaine synthase activity, which could possibly mediate this reaction at lower, more physiological pH. Okay. So we were able to observe salutaridinyl production. It also has the same molecular weight as reticulene, so it appears as a second peak here. And we found that in general we had to codon optimize the P. bracteatum SAL-R, which is more stable than the P. somniferum, even though they only differ by 13 amino acids. So with that codon optimized and high-level expression really of all these downstream enzymes beyond reticulene, we're able to get slight accumulation, but I think there's still some issues with this enzyme being limited by its activity. SAL-R, for example, is substrate inhibited at low salutaridine concentrations, which happens to be the case in most of our yeast strains. It's also NAD pH dependent, so at some point we're probably going to have to go back and look at the overall redox balance of the cell. However, for now we are able to show salutaridinyl production and again show the correct fragmentation patterns. Okay, so for thebaine production, which is really the goal of this pathway so far, we tested the SAL-AT activity. So one of the first things that we did was we codon optimized the SAL-AT since we weren't able to observe the native sequence using Western blotting. And although we see a huge increase in expression, we didn't see a huge increase in activity. So that was a little disheartening. But we were able to play with the pH a little bit, and we do see increased production at pH 7, and we can't really push the yeast beyond that, both because of growth limitations and because we lose the activity of our previous enzymes. So right now we're working to develop a two-stage process, which may require growth and expression of SAL-AT and then increasing the pH so that we can actually get better conversion to thebaine. So right now we do produce detectable levels of the precursor salutaridinyl 7-O-acetate, but it's still a little less than 10 mg per liter. So we definitely want to improve on that. Okay. But ultimately, the goal is to not use norlaudinosilene as a substrate. We want to be producing these molecules and synthesizing the BIA backbone inside the yeast host. So previously, they had published reactions both in Aspergillus and E. coli, converting dopamine to norlaudinosilene. So initially, we found that this required greater than 100 millimolar dopamine in our yeast host, which is terribly inefficient. Dopamine is auto-oxidized, especially at acidic pH. So this is one limitation, but we also looked at transport limitations, and we found that overexpressing a mutant amino acid permease in yeast gave us about a 6 to 7-fold increase in norlaudinosilene production. Also, conversion of the acetaldehyde intermediate is a problem since this is very unstable in the yeast host, as it wants to go to tyrosol or 4-hydroxyphenyl acetic acid, or in this case 3,4. So for this, we were able to test a variety of single knockouts, and we found that we could increase conversion to norlanosline up to 35-fold. And eventually, we want to test the combinatorial knockouts as well and hope that this effect will be additive. Okay. So ultimately, we want to move even past feeding dopamine and do the de novo biosynthesis from tyrosine. So we did some bioprospecting, and we developed a couple of different pathways to do this in the yeast host, both to produce norcochlorine and norlanosline as the intermediate. So norlanosline allows us to move along the pathway as we have been doing, whereas norcochlorine will require an additional P450 reductase activity to give us reticulane. But one advantage of producing the true intermediate norcochlorine is that it will allow access to other intermediates that are not derived from reticulane, such as the family of bis-benzyl isoquinoline alkaloids. Okay. So in conclusion, we've done a lot of work engineering these yeast strains to produce nearly all types of benzyl isoquinoline alkaloids, or BIAs. We've demonstrated the combinatorial testing of different enzyme variants and shown that this can indeed have an effect on your overall production. We've also introduced a novel tuning strategy using the GAL promoter so that you can finally tune enzyme expression levels. And we've also done some work to optimize the expression and activity of various P450s in our system, although this is still limiting in several steps. So the ultimate goal is to put all of these different pieces together. However, we still need some additional optimization, particularly on the upstream part of the pathway. And there's also parts of the downstream pathway that will need to be optimized. But really, if it's moving towards industrial production, there will be a lot more resources to really get that optimization up to commercial levels. But we have demonstrated production. So ultimately, we want to have strains to perform the total biosynthesis of complex BIAs all the way from tyrosine, which again will require putting the different pieces of the pathway together, as I've demonstrated. And a combination of in vivo conversion and chemical synthesis methods on this type of pathway will be very important for drug discovery and eventual manufacturing processes. So I'd like to thank the Smolky Lab and Peter Ficini, Peter Gungrich, and Dennis Pompom for contributing plasmids and yeast strains to this work and funding the Caltech Center for Biological Circuit Design and NIH.
So beautiful presentation. We have time for a couple of questions. So, may I ask the first one? You have been trying to optimize activities of your enzymes by, for example, codon optimization.
We've done codon optimization.
For obvious reasons, I like a lot. Nevertheless, did you ever try to do some approaches like directed evolution, sequential?
We haven't yet, but I think
permuting?
that's going to be required. So.
Okay. Any other questions, please?
Yeah, my question is, with these anti-cancer properties, is it known what the target of these compounds is at the moment?
Actually, the most I've read is that they know that some of them have antioxidative effects.
Okay, and in many cases when you have a pathway that's making complicated molecules, the intermediates in the pathway also have some activity.
Yes.
How do you know that your yeast, that you're trying to overexpress these pathways, is not itself susceptible in some way to these compounds?
Actually, that is a concern, especially with the antimicrobial agents, but we haven't observed any growth retardation at all to this point. But that's definitely a concern with some of our products, and we expect some of them to be toxic to the cells.
Any further questions? If this is not the case, I would like to thank the audience for the discussion, for the contributions.